Ladies and gentlemen, 2017 will be an extremely exciting year for Biomarck. Doctors Adler and Dixon will provide you more detail and information in their presentations. But as you are aware, we have 2 upcoming clinical trials starting soon. These trials are extremely important to Biomarck and also more so to the patients who we will be treating. Potentially the people in this room yourselves and other absent investors of Biomarck might be responsible for saving even 1 or many lives this year alone. Our first presenter whose shoulders Biomarck stands on not only is pivotal to our success, but also continues to be an ambassador for our company. Dr. Adler's awards and achievements are many, and I can't mention them all today, but please allow me to say just one that he received a week ago today. North Carolina State gave him the Research Leadership Academy and 2016-17 Alumni Association Outstanding Research Award from NC State. Ladies and gentlemen, Dr. Adler will be given a medal on this award April 26th from NC State. But please join me in welcoming him to the stage and congratulating him on this achievement.
Thank you, sir. Is there a pointer?
No pointer, but forward and back.
Say it again?
Forward. Yeah, green is forward. And red is back.
Okay.
Your slides will play automatically.
Well, thank you, Rajan. Indeed, I was just notified last week about this award. It's from the Alumni Association of North Carolina State University. I'm very pleased to be honored in this manner. So what I thought I would do this morning is sort of update people on the work going on in my laboratory. For the last few years, when we've come to these shareholder meetings, I presented the research that I'm doing. Sometimes went on a little too long because, as you can tell, I'm very enthusiastic and excited about the work that we're doing in both cancer and acute lung injury and ARDS. So what I thought I would do this morning is sort of go back over what we've done in the last year and how that relates to the clinical trials that are going to be ongoing this year. So let me talk about cancer first. I guess the last couple of years I've shown you all the work that we've done with the mouse models of lung cancer. We can induce essentially the same kind of lung cancer in mice that we see in humans. The tumors form in the lung, they metastasize to different organs in the body, and giving the drug BIO-11006 by inhalation will stop the metastasis and stop the growth of the primary tumor in these mouse models. And as I've presented previously, we can even start treatment of these mice several weeks into the generation of their lung tumors at a time when there's extensive metastasis. This essentially recreates a patient presenting to a doctor with lung cancer. Usually those patients already have pretty widespread metastasis. So what I did over the last year is something that I was really concentrating on was what's going on in these cancer cells. We can tell that that 11006 stops the cells from moving, so I thought I'd look at these cells in what's called in vitro, in plastic chambers. Take human cancer cells, grow them in plastic chambers, plastic dishes in a laboratory, and these are cancer cells. So these are models of metastasis. The cells migrate, they move on these dishes similar to the way they do in the body. And what we do is treat these cells with 006, and lo and behold, the drug stops these cells from moving. Okay, this is great. We know it works in mouse models. We're going to see very soon that it's going to work in humans also. The question in my mind, being a basic researcher, is what's going on in these cells? How does 11006 stop these cells from moving? And I'm a cell biologist. That's how I was trained. So my interest always is what's going on in these cells? How is this drug stopping these cells from moving? So what we do is grow cancer cells in these dishes. These are called transwell inserts. We grow the cells in the little insert on the right, we put them in these chambers. We add growth medium to keep the cells alive and functioning. And we can measure how these cells migrate from that dish on the right down through a membrane, which is similar to going through a blood vessel, and actually look at these cells, at the mechanisms within these cells. Why are they moving? What is it about these cells that causes them to move? And more importantly, what is it about 11006 that stops these cells from moving? So this is what these look like. I don't think I've ever showed you this before. You've seen graphs showing that the drug stops the cells from moving, but this is an actual picture of cancer cells, human lung cancer cells. The cells are called PC9 cells. These are cells that were taken from a very, very aggressive human adenocarcinoma, lung cancer. When we put these cells into these chambers, as you can see on the left, they migrate right through those chambers. And what we do is stain them with a red stain, and you can see if the cells are untreated after 12 hours, they've already gone through that chamber into the next chamber. So the cells are stained. You can see all those cells there that have migrated. The cells on the right are cells that are treated with 11006, and you can see very, very sparse migration. It cuts it down to like 95%, which is the same thing we see in the mouse models of human lung cancer. So it's very, very effective treatment. And as I said, let's take a look at these cells and see what's going on. So before I show you that, I want to talk a little bit, I'm sorry for being a professor here, but there's a protein called actin. If I do this with my arm, there's motion, there's movement. And what's happening is this protein, actin, is part of the muscle. And I won't go into it in great detail how it works, but without actin, you don't get movement. Now, interestingly enough, the same protein that we see in our muscles is also in cells. Cancer cells have a lot of actin because that's how they migrate, that's how they move. So in order for a cell to move, the actin, similar to my arm moving, the actin has to interact with other proteins in the cell, just like it does in the muscle, and cause the cells to sort of move. You've all seen pictures. I guess people here are old enough to remember a movie called The Blob, which is actually Steve McQueen's first movie from, I think, 1956, 57. And basically there was this blob, hence the name, and the way it moved was to send out this like big processes and grab on and just keep moving like that. And I'm sure if we did a cross-section through the blob, you'd see all this actin. Note to myself, get hold of the blob for me. Okay, and what we see with 006 is 006 directly blocks the function of actin in these cells. It causes a rearrangement of actin. And when the actin gets rearranged, it can't interact with the proteins it normally does, and the cells don't move. So how do we know this? So I want to present this picture to you, which is, I think, in my mind, one of the most important things I've achieved in my laboratory. So these are the same cells that you saw with the red stain, but now we're looking at them a different way. Now we're looking at them through a microscope called a fluorescent microscope. And the way this works is we take a dye, a fluorescent dye, something which under the microscope will shine amazingly fluorescently with a green dye. Green dye is called fluorescein. And so everything you see here stained green is actin, the protein actin. So what's interesting, if we look on the left here, these are cells, these are cancer cells, the same PC9, that very, very aggressive human cancer cell. And you see all the actin, I don't have a pointer, but all the actin here is around the cell. All the actin is around the cell. It's a cross-section, which means it's on the outside. Remember the blob, the cells are moving. Okay, we treat them with 006 and look a few hours later, and lo and behold, the cells totally change the way they look and what happens to the actin. So what you see on the right are three actual phenomena related to a 006. The first thing is the cells go from a rounded configuration, and I keep going back to the blob. The blob was round and kept moving. They flatten out. This is the same magnification. And you can see the difference between the cells delineated by the actin on the outside on the left and the cells on the right. So they flatten. Cells that are flat don't move. Second thing, look what happens to the actin. I want to point out two things, and you can see from the arrows. One is the actin, instead of being on the outside, now is through the middle of the cell and traverses the cell. What these are called in the cell biology world are called actin stress fibers. And what they do, they're very important for cells that don't move to adhere. So rather than moving, these cells are now adhering to their substrate. Substrate in here is the dish that I showed you. In the body, they would be adhering to either the matrix that they're on or whatever tissue they're associated with. And the second thing, and I'm going to walk over to the screen and show you these because this was one of the major findings of this study. You see these features almost look like foot-like projections. You don't see any of those in the normal cancer cells. You treat them with 006, these features become apparent. So these are the stress fibers I was talking about before, which are traversing the cell. These features are called focal adhesions. What these focal adhesions do is serve as anchors for the cell. They grab onto that substrate and they don't move. This is characteristic of cells that adhere and don't move. So basically what we're seeing here is that 006 changes these cells from cells that are very, very motile, cells that can move very easily, to cells that are now grabbing on to the substrate. We're going to see a presentation by Raymond Rodriguez-Torres. One of the interests we also have in the laboratory is pediatric cancer, not just lung cancer. Live Like Bella, you're familiar with Raymond's daughter Bella, who died of a disease called rhabdomyosarcoma. This is a terrible cancer that affects mostly children, and we found that 006 also has the same effect on these rhabdomyosarcoma cells as it does on the lung cancer cells, and we're very anxious to get started with Raymond and doing some work in pediatric cancer. So I'll show you the same pictures now with rhabdomyosarcoma cells. The cell line is called RH30. It's very, very similar to the rhabdomyosarcoma cancer that Bella Rodriguez-Torres had. And again, you can see going through that same dish, on the left are those cells that are untreated, very, very metastatic, motile, moving cells. When they're untreated, when we treat with 006 on the right, you can see very, very few cells are migrating. So it works, seems to work in pediatric cancer as well as in lung cancer. And when we look at the cells through the same confocal or fluorescent microscope, you can see the same sorts of effects on the pediatric cancer cells that we saw on the human lung cancer cells. On the left are the normal cells. These, rather than being round or sort of spindle-shaped, these rhabdomyosarcoma means it's derived from a muscle, So these are almost like muscle cells that you see, sort of spindle-shaped. But again, all the actin, or most of the actin, seems to be on the outside of these cells. And treat them with 006 and the same features become manifest. The cells flatten, change their shape. They express the stress fibers, and you see some of the focal adhesions. So we're very anxious to get started looking at some pediatric cancers. We've looked at other cancers, osteosarcoma, glioma, brain cancer, and the drug has the same effect on these cells. This has all been done in dishes and chambers, hasn't been done yet in animal models. Yeah, neuroblastoma is another disease. So whatever the drug is doing to cancer cells, it seems to be something that occurs in a variety of different cancers. And we're very excited to be starting to do some animal work with cancers, animal models of pediatric cancer, and test the effects of 006. This could be very important. I don't know if you remember Raymond's presentation, but there's very, very little research being done in pediatric cancers, and it's a shame because they're just it's just an awful thing to happen when a child gets sick like that. Okay, so I'm going to finish talking about the ARDS work, and trials are underway at UNC. Dr. Wayne Anderson in the audience is involved in the clinical trials with ARDS. We published last November a sort of groundbreaking paper in the American Journal of Respiratory Cell and Molecular Biology, and showed that 006 can reverse acute lung injury in mice. This was unknown. This was the first time any drug. There are some drugs out there that can prevent it. If you give the mice the drug, then hit them with different stimuli, which will cause the equivalent of human ARDS, it can block it, but nothing works like 006 in reversing the acute lung injury and acute respiratory distress syndrome. And when we have patients in the clinical trial, they already have the disease well established, and we're pretty confident that the drug will reverse the disease process also in these humans in the clinical trial. So I'm just going to end. You've all seen the video previously, little snippets of it, the mice treated with the drug. So what I did was take the entire video, it's less than a minute, and it shows the mice actually being treated with the drug and then the effect of it. So this is actually the entire director's cut here, which the previous ones were derived from. So these are the mice. They have the disease. This is like 24 hours into stimulus. You can see how awful they are. They're trouble breathing, hair is standing up, eyes are closed, lying in their own urine, no movement, rapid breathing. Left by themselves, they'd probably die within another 24 hours. Here is our exotic chamber. Okay, you see the nebulizer there. You see the glass top, and there are the mice inside the chamber as soon as we take the top off. And you can see how happy they are. They just love this drug. And lo and behold, they've been given the drug for half an hour. And this is the same mice. This is what they look like.
Amazing.
Bright-eyed, bushy-tailed, very excited about their newfound drug effects. So that's what I had to present. I thought you'd like to see the real untouched video of this. As I said before, clinical trials are underway or will be within a few weeks in both ARDS and cancer.